Well, welcome everyone. I'm Paul Danis with the consortium here, and we're glad to have you join us for today's Proteoform Thursday. We're excited to have Jeff Agar with us from Northeastern. Should be an interesting talk about something a little more applied than what typically some of the basic work is, but it's definitely going to delve into some areas we haven't seen before. It should be pretty exciting to hear about. So I look forward to hearing the story, Jeff. Just to maybe give a couple quick updates here. First, I'd like to have
Wait,
a thank.
Paul, you might
Yeah.
want to take a look. I think I hit the wrong button, put everybody in a waiting room. I'm so sorry. What have I done? Okay, good. People are still here.
Okay. Thanks, Jeff.
Paul, can you check the meeting? I don't see. I think there's a big giant waiting room without. I don't or not. I don't know.
It looks like everyone's here.
Okay, good. All right. So is it? So am I taking it now and going?
No, I have to give an introduction first,
Okay,
Jeff.
perfect. All right. I'm so
All
confused.
right. Thanks. Welcome everyone to Amateur Hour here at the Proteoform Thursday. Let me so great to have Jeff here. And let me just give a quick intro on a few things. First off, we want to thank our sponsors who enable us to do these types of things. Our major sponsors have been with us for several years: Thermo Fisher, Brooker, Cyax, and Lilly. Very grateful for their support. And additionally, our supporters of Agilent, Pfizer, and Mass Spec Consulting, or MSC, as well as welcome to our new sponsors, New Objective and Impro. So we really appreciate what they can do for us, and we're happy to have them part of the community. If you're part of an organization that would like to support the consortium, please contact myself or any of the people involved in the consortium, or you can do it through our website. And we'd love to have you involved and have you supporting us as well. So the big news is that as of today, registration is now open for the third International Top-Down Proteomics Symposium. It's to be held in August in Hamburg, Germany, which should be an exciting time. It's going to have a range of basic information on technologies, platforms, applications, and we look forward to seeing a lot of you folks there in August. So you can go to tdp2025.org if you want more information and you want to start your registration process. Additionally, we have quite a range of speakers already lined up, from our esteemed speaker today to Albert Heck, Neil Kelleher, Ole Jensen, Charlotte Utrecht, Liliana Pastolik. So a wide range of great speakers here from all over the globe. And one thing we're actually implementing this time as well is we're having short courses. We realize that one of the challenges with top-down proteomics is education and becoming comfortable with these complex workflows. So we have two sets of courses available to help people with this. The first course is a complete overview of all the workflow and all the details of how to operate this, as well as some of the specific applications and biotherapeutics, as well as disease proteoforms. The instructors are outstanding, some of the leaders in the field, so this should be a great course. And this is just before the conference on the Monday and Tuesday. In addition, a post-conference workshop we have on the data applications. We know those are one of the challenges, is trying to extract useful information out of proteoform data. So we'll have this course as well for one day, basically focusing on the identification as well as quantification of the proteoforms. So that should be also valuable. So you can sign up for those as well on the website. And it should be a fun time. Hamburg is an exciting city on the port side here. So please take a look, join us, and hope to see you more there. We'll also have information about abstract submissions coming out shortly, and we'll let you know as soon as that's available. And another cool piece of news is we have Philip Kaulik from Kiel University be talking next month. He's been talking about some of the truncated proteoforms that he's been studying there for a while. That should be another interesting thing. So we'll have information and details of registration coming out shortly on that as well. So finally, let me turn it over to Jeff Agar. Jeff's Associate Professor, Chemistry and Chemical Biology. He's also at the Barnard Institute of Chemical and Biological Analysis and is the Associate Dean of Professional Programs at Northeastern University. So let me hand it over to you, Jeff. Take it away, and we look forward to hearing your story. And there'll be questions and answers afterwards. But if you want, you can just drop something in the Q&A if it comes up during the talk, and we're happy to address that as well. Go ahead, Jeff. It's all yours.
Okay, thanks, Paul. You might want to check the waiting room. There's one person in there I couldn't wrench out.
Okay, I'll get that.
I'm sorry, brother. I did. We do our best.
Thanks.
Right. So let's just find. Welcome, everybody. It's a great list. So, little warning. This is the first time I've given this talk. It's not, most of it's just published or not published. And I thought it was going to be a 45-minute talk because I wasn't listening. And it's 30. So I'm going to try and get through this quickly. And I'll skip a few things. But this is about an intact protein mass assay. And it's really about how you use that to monitor covalent drugs and how they get through the body. And when Paul said it was applied, it really hurt me because from a pharmacological perspective, we're going to show you that we derived a bunch. It's the first thing that I've ever had for in vivo bioanalysis of covalent drugs that had a mathematical model behind it. So hopefully will be more than just applied. I guess I shouldn't be offended by that. I just get I'm so sensitive, Paul. So let's start with this. We have, I just want to kind of couch this way. Medicines have been around for a long time, as long as we've had recorded history. Birch bark, you could chew on it, and then you could get some version of aspirin, salicylic acid, and then acetylate it, and you had aspirin. And that was 1900. And then you started making kinase inhibitors. What's fun about everything I'm going to show you here, people said it was impossible and it wouldn't work at the time. So kinase inhibitors were not supposed to work, and they became the blockbuster drugs. Structure-based drug design happened. And I'm just putting these times, of course, they happen. Academics are always leading in this before, but when they're adopted. And then finally, I've lost control. I'm sorry, I'm not able to advance my slides. Give me just a second here. There's somewhere I could put my cursor, Paul, where my slides will advance. I'll start over. By the way, can somebody just say that you hear, take your mic, your audio on, and say that you hear me? Because this is like dead silent.
Yeah, Jeff, you're fine, Jeff.
Okay, thank you.
Everything's fine. Your slides look great.
No, Paul, it's not fine. Okay, all right. Anyway, so then we make intentionally covalent drugs, probably around 2015. And oh my gosh, my slides won't advance. There we go. Okay, good. All right. So here's where we were. Make this fast. Somebody made covalent drugs. We're going to stay in our lane here of small molecules. There's a bunch of stuff that you all have done, and it's really cool with the big proteins and advanced therapeutics we don't have time to get to. Unfortunately, I'm going to have to tell you about the first 10 slides, which is what we did starting in the modern drug era to make sure drugs were safe before you understand why what we did was necessary. Okay. So I want to, I'm an inorganic chemist. I did a PhD with spectroscopy, a little postdoc in mass spec, and another one in neurotoxicology. I'm not formally trained to develop drugs. So why I'm going to get up here and be able to tell you this is because I was teaching a bunch of courses to locals, continuing education in Boston, and they were going from small molecule analysis like the one I'm doing here and wanted to learn how to do intact proteins. And I had an aha moment when I went, wow, there are a lot of really smart people in this room. I'm going to start recruiting them. We're going to spin up an industry PhD program so that they can either keep working in industry and work in the lab, or maybe we can just get them right out of industry and into the PhD program. So these are the people, and they deserve special thanks for this. The companies here aren't the companies that they work at now. Those are the companies they worked at before they entered the lab or while they lived in the lab. If they're highlighted, it's because they actually did work in industry the whole time through. So our lab was special, and it's a place where you can sit across the table and have most of big pharma represented as you try to develop a drug and keep an academic out of trouble. So thank you all, everyone on this list. The work that I'm going to tell you is each one of these people contributed to. Okay, let's start with the basics. Why do drugs fail clinical trials? Academics, and it used to be this way in pharma, really just think about potency. How low can you get the Ki or the Kd of your drug, right? But then Chris Lipinski came around and said, we're failing way too many clinical trials. And he came up with Lipinski's rules, which some of you may have heard of. If you didn't, it's been cited last I checked like 9,000 times. It's a very important paper. And he showed that the physical chemical properties of the proteins alone in a meta-analysis could have explained why things didn't make it through clinical trials. And those physical chemical properties, hydrophobicity, a number of hydrogen bond acceptors and things, affect especially adsorption, distribution, metabolism, excretion, and not so much tox, but I've written it there so that when you see ADME what it means. And so now it's just common practice to co-optimize the potency with the ADME. You can't do one without the other. And everything we're talking about today, let's be clear, is ADME. And I do think, this will be interesting. We have some instrument manufacturers in here who can comment on this, but I was always surprised to go into like MDS Pharma Services or different places and see just how many triple quads there were. We don't read about it as much in the literature, but it's a big part of what's going on, and they're quantifying drugs. So what makes a compound drug-like? Those are Lipinski's rules. It's a rule of 5. It's these parameters. We're not going to go over them, but it has to be small and soluble and not give or make too many hydrogen bonds. And it will then affect things like whether it's orally bioavailable, whether it goes into one from the blood to the brain. Well, let's leave brain aside for now, but to the liver and things like that, how it's metabolized. Lipinski's rules don't tell us this. They don't tell us anything about blood-brain barrier penetration. There's some relationship, but there are different parameters that play there. And it turns out in other meta-analysis, inability to cross the blood-brain barrier is what led to the catastrophic failure rate we have in neurological disorders, which is over 95%. And thank God for MS, which has a peripheral component. By the way, I mean multiple sclerosis. Because of that, we're doing better in clinical trials. And Lipinski's rules don't really help us with covalent drugs either. So we have to know these basic things to understand this talk. One is pharmacokinetics, and this is what the body does to the drug. This was the big missing factor. We need to know the pharmacodynamics, what the drug does to the body. That's the point after all. You combine PD and PK and you have pharmacology. And the central tenet of pharmacology is that the magnitude of the response is proportional to exposure or concentration at the site of action. Okay? And this is the rationale for our studies. This central tenet, and this really wasn't recognized as well as it should have been for covalent drugs, this central tenet is predicated on reversible binding. If you follow the math back, every assumption requires this, otherwise it's not true. So this I want to point out, everything that they're doing with traditional PK requires a lot of extrapolations. Extrapolations, all you're doing is measuring drug concentration as a function of time. To do something with that, you need to do some biochemical studies, establish a KD or a Ki of your protein. You need to do some cell studies and establish a dose response. You need to try to put them together with the Chen-Prusoff equation. And there's a lot of extrapolating going on. And if you remember, one thing about what we're going to do is the trick is we just cut to the chase and we just measure percent occupancy. So that's the basis of this, is to just cut through these things because we can with the mass spectrometer, right? We can see the bound and the unbound part, and I don't think anyone does it better. This is traditional PK. In blue are the data, right? You're just going to get drug concentration. From that, a set of parameters that are now necessary to get a drug approved are going to come out. Most important, probably, what's your maximum concentration? That's very important. Somebody else is going to figure out, look at this, where you kill an animal, right? There's going to be a toxic level. You're going to be absorbed from, let's say, the blood into the rest of the body. And now your body is going to start acting on it when you're absorbed, and you're also going to be distributed into the organs. And then after that happens, your liver is going to start beating the crap out of it and metabolizing it. And that's this phase. And then you're going to be eliminating it, maybe excreting it through the kidneys. Maybe you forego the liver. I don't want to simplify things. And then you have to have a minimum effective concentration, right? This defines the therapeutic window, minimum effective concentration, right? And we have some drugs, like for cancers, that have just like a two-fold window, right? That you can really do a lot of damage. So this works with small molecules so long as they're reversible, non-covalent. You could take the ratios of two of these area under the curves, which are also important, for, say, two different routes of administration, IV, oral, or for two different tissues. That would give you the absorption and the partition distribution coefficients. So right here in one slide, and it's fast, I know that, you have all of the basic pharmacokinetic parameters fall out of this. And they're modeled in a pretty straightforward way, believe it or not. You know, you can predict the concentration at any given time with some constant. It could be normalized. It could be a concentration at a particular time, e to the minus kt, right? That's it. And this will be, obviously, sometimes you'll throw in another one of these terms, but that's the basic modeling. And when you're done with this, you can determine a dose regimen. So now we're doing lots of these little studies. This is 3 different twice per day doses, and you can watch the concentration of the drug, and it does a sawtooth as it's absorbed and as it's metabolized, right? This is what drugs look like in your body, some version of this. Just to point out there, this is the kind of history of covalent drugs. They were avoided by big pharma because they can be haptogenetic. They were literally avoided until recently. But when you were using, and they avoided them in structure-based rational drug design. But there were a bunch of other people just doing phenotypic assays, whether it be traditional medicine or just looking if you can save a cell. And through that, some cool things happen, right? Penicillin, Prilosec and derivatives thereof, and a lot of things. And then recently it changed. Somebody realized that you could take a kinase inhibitor that wasn't selective, and to that you could append an electrophile, like an acrylamide, to bind to a cysteine that's only at one of your kinases. And that's what all of these are. So they call that an affinity of first approach to covalent drugs, okay? And then people just said, heck, if that's working, why don't we just take large libraries of electrophiles? So this is an electrophile first approach. Why don't we use chemoproteomics techniques like Ben Cravat, Steve Geigy's done some really interesting, a lot of people have done this. Now we just put in electrophiles, we find out where they bind. And when you do that, you get Amgen's blockbuster, Ceradicib. You get the main ingredient in Paxlovid here. And today, I'm going to show you results from these 3 drugs with using our assays. 2 of the 3 worked. Okay. So this is what happens, though, if you have a non-covalent drug. This is the free drug concentration, just this little tiny little blip in time. And this is the effect on target engagement. This uncouples pharmacokinetics and pharmacodynamics, and then there wasn't a solution for this, and this is what we're trying to solve. So we measure this directly. And this is what it looks like, you know, a real study. You do all these different concentrations. This is obviously a reversible binder, and you go from drug concentration to target occupancy. And I've said this already. I'm just going to gloss over this, but the best thing that they had in industry for covalent drugs was actually an in vitro measurement. And it's basically just measuring, you have an affinity constant here for the reversible step, and then you had a rate for the formation of that bond, okay? That's all they had. It's called Ki over Kinact, and we're gonna do something more than that. So this is, I'm gonna have to gloss over this. The early years of my lab were just figuring out what actually causes familial ALS. Its aggregation and its loss of protein stability. We didn't need mass spec to do that. That was just epidemiology. Then we started doing mass spec of the ALS patients, and this is kind of a proteoform profile. A lot of stuff we didn't understand, but and you could repeat it and you see all these proteoforms. And so the take home is this. We learned in vitro to take the protein, we could add cysteine, and then that would make this proteoform, okay? And then we could just add the enzyme's own product, the thing that it makes, right? And that'd be peroxide. And now this gets modified to 2 and 3 oxygens. Really amazing how specific it was. Turns out to be on one residue. And this one was protected. And in doing that, we learned, wow, in vitro, we can now recreate the picture that we see in patients. And it tells us that there's a single cysteine that's being oxidized, and your body has a protective mechanism via acetylation or cysteinylation. It only happens in nervous tissue as far as we can tell. But that's just correlative. It doesn't mean anything. So we learned how to take this one proteoform and make it and purify it. And that's this proteoform. And these are 15-year-old ECD data we used to be proud of. They were never really great, but at least we did some top down and found out where it was modified. What's important though is we made this proteoform a single residue of all the 153 modified by 3 oxygen, and we gave it to experts, and it slows exonal transport, and it's as toxic. It's a wild type protein that's as toxic as any mutant. And then a bunch of other people, we were like, hey, go make some rescue mutants. And they did. And so basically they could take that cysteine, mutate it to a serine, and it made animals live longer. It made cells live longer. Okay. So now as a field, we have like, wow, we got to stop this cysteine from being oxidized. And also other people's work showed that this dimer, once you get an ALS causing mutation or once you oxidize the cysteine, falls apart. So we have to, if we can stop the 2 most important things, if we can stop that cysteine from being oxidized. So that's the old part. This is the new part of the talk. We're not the only ones trying to do this. A bunch of other great ideas came out where they're like, hey, we have different approaches for doing what I just said, blocking that cysteine from oxidation and holding the dimer together. So our biological goal as ALS researchers is just to evaluate these candidates. We can't do that. There weren't techniques, so we'd have to enable this bioanalysis. So we created these cyclic thiosulfonate crosslinkers, and they're just a crosslinker. The only thing that's different is that I imbued them with a sensing mechanism. So if this is not my target, maybe it's a cysteine protease, and now it binds. If it's a normal crosslinker, that bond is made and it's very toxic. Ours has entropically driven ring closure to reverse this reaction. And if there's a second cysteine present, now we left an oxygen there, you can have water as a leaving group and a big driving force. So that's our chemistry in a nutshell. This has been published, and so in the interest of time, this was published last year. I'm just going to really scream through it, but I refer you to this. The most important thing is that these really stabilized ALS causing variants across the board. They turned monomers into dimers. This one surprised me. Without us even putting metals back, they turn inactive mutants back into active mutants. We were very happy about that, or variants. And somebody solved the crystal structure. Thank you, Samar Hasnain. And as a mass spectrometrist, I just go, wow, that looks nice. So now to the part of the talk where we're going to develop bioanalytical methods to now start evaluating covalent drugs. This isn't, again, we're not just solving a problem for ALS. This is across the field. There wasn't an in vivo technique that I'm aware of that could do any of this. This was just published last month, and probably some of this that I'm going to show you is in there. First thing we did is just do bioanalytical methods development. For those of you who want to start, this is a good place. The methods really don't go that far from what we published here. I want to point out, everybody who's doing top-down, please, if you have thiolation, whether it be glutathione, cysteine, any type of thiolation, a lot happens in your sample after you lyse it. So go anaerobic or add blocking agents, and that's what we were doing here. It turned out for our drug-bound, I'm going to say drug, of course, it's not a drug yet, but for our drug-bound complex, it turns out not to be labile to these sorts of modifications. So we didn't have to block in the final method, but at least we looked. So now what we have at this point is we take a drop of blood. We do a quick solvent extraction, chloroform-ethanol, and we just inject that. We get a monomer, we get a dimer. This happens to be ubiquitin, still sticks around, always ubiquitin. And we can measure the relative amounts of monomer and dimer, get a ratio of target engagement, which is what this is. And for us, the ratio of target engagement is the pharmacological effect. We had to hit a threshold of 37% to be as good as the best thing that exists in ALS, which is RNA silencing. It's hard to develop drugs. It takes a lot of expertise. And so the team put together a decision tree for others. So you don't have to go and hire a bunch of people. You can just sort of follow the instructions. And I'm not going to go through all of this. I'll go through it as the talk proceeds. But basically, you ask, does your drug bind the target with the MOA when it's in vitro? Can you hit the target engagement threshold that you need to have an effect? Can you do that in a tissue homogenate, the tissue that you're trying to target? And from that, if you do that in a concentration-dependent way, you can actually extrapolate the dose that you need for animals. Then you have to kill some animals and find out what the LD50 is and ask, does the dose that I think I need, is it going to kill the animal? And if it will, you stop there. So this is kind of the idea. So let's go through it. Here's the papers that I showed you earlier, the leading candidates for familial ALS. These are all from other groups. This one's from ours. Is the MOA correct? Well, gotta love mass spec. Every single one of them bound where they were supposed to. There is a lot of top-down behind this as well. They bound where they were supposed to and by the right mechanism of action. Did they engage at least 37% of the target? Yes, they all engaged 100%. Getting to this, I want to point out the power of mass spec. Getting to these molecules was not easy. We did a computational screen of 200,000 compounds, and we screened every single published 3D dimensional, 3D protein structure. Because if the motif that I was going after here with these two cysteines, it shouldn't be all over the place. Cysteine is only 1% of amino acids. So two cysteines next to each other by chance is 1 in 1,000 occurrence. But, you know, biology has a tendency to make motifs. But we didn't find that motif in a lot of proteins. And so we proceeded. We got 154 compounds with this correct scaffold that we thought would work. 83 of those formed dimer. But this is where it gets really cool. Only 29 of those use the right mechanism of action. And so look in the upper right of the screen, we're trying to do a nucleophilic attack on this sulfur, which will then open a ring. But if this, we learned through these studies that if this carbon becomes electrophilic enough, you attack here instead. And if you do, that's the wrong mechanism of action. And if you do, it leads to the release of persulfide, and you get this god-awful SOD1, with the two cysteines you're targeting and a polysulfane sulfur linkage. To me is amazing. John Hart solved this crystal structure. And by the way, this is the same crap that Sigma sells you. So if you buy SOD1 from Sigma, most of it actually happens to be in this proteoform. Turns out it doesn't come from our types of drugs. It comes from the dialysis bags. To me, is amazing. Just love having mass spec there to, you know, keep us honest. We could do this we could buy a bunch of proteins from our computational screen and get a vague idea of selectivity. That looked okay. We could then do this in tissue homogenates, okay? And ask, can we reach the minimum effective target engagement? And we could for 3 of the 4, but cisplatin died at this point. We could extrapolate the EC50 in dose through studies like this. This is just a tissue homogenate, different amounts. And if you look at where 50% is bound, it would extrapolate down to these doses, 13 mg per kg in brain and 67. This is going to be a lot of data here, but it's important. Our 7-membered rings killed animals they're ridiculously toxic. Our 5 and our 6 weren't. And this is intravenous, subcutaneous, oral LD50s. And these are a bunch of compounds in cells. The good news is that we were under the LD50, and so we could proceed. This, look here, are our 3rd generation compounds. We now have things that have mg per kg toxicities that I won't talk about more than just as a teaser. So this was a go. So I'm going to skip the spectra to show you that we can, but these are just, this is a monomer untreated. This is the actual experiment that gave us this, and this is from in vivo. So they match pretty well. We were really pleased when now we put these into animals and look, and brain was really good. Brain was just what we thought. It was amazing how well it worked. From the extrapolation from a tissue homogenate to an actual animal's brain was just what we thought. Blood did better than we thought. And it turned out that that was because our drug has a low volume of distribution. It stayed in blood. So we would learn that later, but it's interesting. So we got a yes here. We could do this in animals. But everything else failed at this point. Disulfiram, Epsilon didn't make it. So this was what made it through. And we get these curves, red blood cells, percent dimer as a function of time, and they're great. But what do they mean? And I want to tell you, we started doing animal studies at this point before we had developed the math to interpret these. And I'll show you later that we had no choice because it would take us a couple of years to develop the mathematical framework and do the things we needed to do. But I wish I knew how to do this. I wish I understood this better before we started survival. This is published, you can go through it, but we derived for what we're calling intact protein PKPD. All right, we blew through all the math, and at the end of it, it looks very similar in form to traditional analysis, except the K's mean something fundamentally different. This K is an elimination constant. It's how fast your body metabolizes the small molecule itself. Our K turned out to be protein, the ternary complex of your drug and your protein, and its degradation half-life. They're a protein metabolic, an engaged protein metabolic half-life, which is interesting. We could do it for 2 compartments if we can do it for 1, and it's been working very well. So now this is actually modeled. This is an oral distribution, so you don't do this type of modeling from the oral, but this is IV, and these are actually the parameters that come out. Percent dimer, maximum concentration after 8 hours. We have an AUC. We know our bioavailability. It's 8%. It's not great, but it could be an orally bioavailable drug. And that's way better than what we have right now, which is to stick a needle into their spinal cord and inject it into their CSF, intrathecal. It penetrates the blood-brain barrier, and its effect has 2 half-lives. We don't understand why just yet, but there's an 11-hour and an 87-hour pool of proteins. So this has a pretty prolonged effect in these mice. We would then go on to apply this to, and I point everyone to Neil Kelleher's great work with KRAS, which inspired us, but we could do the same things with KRAS and soraticib, BTK, and ibrutinib, and show that it worked. These are some combination of from in vitro to actual human lung samples for KRAS. And we also saw something cool. A lot of drugs, especially covalent, fail because they never make it past your plasma. And if they don't make it past your plasma, it's probably because they're binding the reactive cysteine in BSA, or your human assay in serum albumin. So we could try these old drugs, and what we saw was really cool. This is unmodified albumin. Doing a quick time check. This is unmodified albumin, and then it gets modified. The older the drug, the more it modifies albumin. So even these kind of affinity first electrophiles, we'd always see a minor adduct, but by the time we get to ceratosid, no adduct. And ours didn't make one either. Now look at this the little asterisk. That's a proteoform of albumin that is cystinylated. In other words, its reactive cysteine is blocked. And that proteoform didn't bind any of these. It was blocked. Well, actually bound cisplatin, which told us that cisplatin targets more than just cysteines. Probably no surprise. So with that, we could look at, by IPKPD, we had all these parameters, but small molecule PK didn't work at all. Our compound goes in and does its thing very quickly. Within minutes, it's bound. So we just had nanomolar concentrations of the compound, and basically the take home would have been your compound will never work. Less than 1% will be engaged. We figured out some release chemistry so that we could unbind the drug and then quantify it, and that's what this is. And even that gave us very different parameters, like 3-hour half-lives. Again, we started these studies before understanding how to deal with our dose response curves. But I was really happy, the mice that we did all of the PK in, G93A, were living a little bit longer. And look at the studies, and these take more than half a year. G85R mice live longer. We then scaled this up, didn't pay attention, and did this in a we took it for granted that this should work in any background. So this was a B6, New Zealand black background. And if you move to an SJL background, it didn't have any survival benefit. This is a big mistake. It's not a mistake, we just didn't know at the time, but even a proxy of this is a $300,000 study. You don't even wait for them to die, you just do NFL levels and see if they go up. It's $300,000. These are more expensive than that. We could now go back and do the IPKPD and see that in these mice, for reasons we still don't understand, the blood-brain barrier penetration is lower, the exposure is much lower, but now we can go back and calculate better dose regimens and make this better. And then that sort of gets to the end where we've done everything I ever hoped to do, which is to bring this to where it was making animals live longer, put a package together for the FDA. And then nobody told me about this thing called the Valley of Death, which is after that happens, you need 3 to 5 million to pay a CRO to do IND enabling studies to get a package to the FDA. So if anybody has 3.5 million for IND studies, let me know. But the good news, if there is good news for us as a lab, is that we really do like to do analytical chemistry too. And we can do a lot more of that. So you'll see a bunch of stuff coming out while we sit in the Valley of Death this year and actually do more hardcore mass spec, such as this. I hope you'll see this coming out soon, frag maps to disambiguate internal fragments. We've been doing a lot on this, Novera alum, looking for a job if anybody wants to hire an expert. But we're basically using fragmentation propensity to decide and disambiguate. But we've done MS3 and ion mobility on each one of these to make sure that the choices we're making were rational. And frag maps, this will be the last slide, can also do difference mapping. And so I'm going to go back to where we started, which is that one cysteine changed by 3 oxygens amidst thousands of other elements that are not changed. And this is the difference map between modified and unmodified. If it's blue, it's a fragment we only see if cysteine 111 is oxidized. If it's red, it's only in the control. And for people who've read Jenny Broad, Jenny's stuff is Vicky Wysocki, Neil Kelleher. We all know, going back in time, that negative acidic residues do charge remote fragmentation. They use their protons to break things proximally, and that's what we see here. So we see the cysteine 111 once it's oxidized, acting like aspartic acid or glutamic acid. So with that, in addition to the other students, I want to make sure we thank the medicinal chemist Roman Manich, who's made a bunch of next generation compounds, and his PhD student Brandon Miller. Some of the major funding here and some of the minor funding at the bottom from people who have helped us with various aspects of regulatory stuff. And with that, I will thank you for your time. Apologize for going a little too fast, and take questions.
Great, thanks, Jeff. Great tour on the drug discovery program you have there. Sounds great. I have a question, Jason Neal. Did you perform off-target profiling, e.g. ABPP proteomic analysis on different compounds to understand broader covalent activity?
Yeah, that's a really good question. So basically, that's asking, are we doing chemoproteomics? And yeah, I'd say right now we are doing it. And at this moment, we've been doing it for about 2 years. We're not great at it, but we're trying. I can only devote one student to this, but we'll have to get this to get it licensed. Like, we've talked to Biogen about licensing this, and they're like, let's do the chemoproteomics. So we have to, there's wrinkles, but anyway, yeah, we're trying. That's the short answer. And it looks good. What we've seen so far looks good.
Great, thanks. I just had a question on the, you mentioned doing top-down to determine the binding sites on these. Can you talk a little bit about that process? Is that a pretty straightforward workflow, or is there still a lot of art and voodoo involved in having confidence that you would sign a site at 100%?
Oh, that's a bigger question. Yeah, because
I mean, you see these things, they look pretty clean, but then there's these little bumps around it. It's like, how do you know?
Yeah,
binding sites.
So we have a couple of papers that are actually written that will get into the in-depth. I think basically, here's the spoiler alert, everyone. Right now, we use PSMs to assign our peaks. PSMs are not designed to assign your peaks. They're designed to correlate your spectrum to a gene or a sequence. You have to follow up all assignments with bona fide peak assignment using distributions, using math, using confidence intervals. If you do that, the ambiguity is less than you would think. But Paul, it's still big enough that right now you have to either make it an assumption, say, I'm going to take, of the two possibilities for a given fragment, I'm going to take the one with the higher fragmentation propensities, the one that should be there. Right now, we've just been doing MS3 on everything to eliminate the ambiguities. It's extremely time consuming, but it works. Let me get to Mike Senko's question. So it's not my rule of 5, it's Chris Lipinski's, and you're right, there are only 4 things. It's so confusing that they call it a rule of 5. The reason they call it a rule of 5 is because everything, 500 molecular weight, 5 hydrogen bond acceptors, 10 hydrogen bond donors, whatever the fifth, what's the 5th one? Oh yeah, log D. They're all multiples of 5, Mike. That's why that's there. That's why they call it that, because it's the rule of 5. It helps you remember, I guess.
So I kind of a general question. If a CRO want to adopt this kind of workflow to actually put it into production, how do you think that process could occur, or is it something they would collaborate with your group to do?
I don't think they have to anymore. They should just read our paper. Not kidding. The whole point of it was if I try to do anything, if anybody ever wonders why I don't publish 20 papers a year, it's we try to do really hard stuff and then democratize. So now if you want to do this, read develop an LCMS assay. The consortium, not just me, but all the consortium members have made this a lot easier for you. And now we give you the math to model it. So, you know, you're done. Just adopt it if that's what you want to do.
Seems to me I've heard that before. Just read the paper, but somehow there's sometimes a little more involved than
Well, you know what? If it's more than that, then we failed in our goal, because our goal was literally to make it doable. And Paul, it's interesting. We heard read the paper a lot more in 2017. It worked a lot better when the consortium got itself together, did the nature methods paper, standardization, and we've done so many other standardization efforts. I do think we're making it easier for people. I really do.
Great, thanks.
But the hard part, I will say this, Paul, this was a big surprise to me. I study neurologic disorders. So whether I'm trying to, and I've been able to, I can take a microliter of tissue and get a signal back from SOD1, tau, alpha-synuclein, ubiquitin, effectively all of the major histopathological markers for Alzheimer, Parkinson, SOD. It's just very easy to get intact protein mass spec of those, whether using antibodies or even now with SOD, we don't use antibody. We just do solvent extraction. When we moved into oncology targets and we did KRAS, it gave me a whole new appreciation for what Neil was able to accomplish. We need 200 mgs of tissue minimum, because that protein is nanomolar, 20 nanomolar or whatever it is. So for those of you who are studying targets that are at very low concentration, and you're going to do this in an animal, you're going to either have to overexpress it in the animal, or to make your life easier at first, which is now cheap and you can do, or you're going to need a lot of tissue. So there you go, Paul. That's the one thing. We give tools for that too, how to figure that out.
That sounds great, Jeff. Sounds great. However, request for position, postdoc looking for a position there, so that's always good to see.
We love that.
Looking to develop. Just a thought out loud is, so you need 3.5 million to move this forward. Are there any opportunities around disease foundations that may have interest in moving this kind of thing forward?
It's really funny. I'm trying. I will just say this, and I'm in all honesty, I've donated my summer salary for probably at least 15 years. I give like what I can give myself. We did these cyclic thiosulfonates. There's a very broad patent for this that I have, and I was able to get that licensed by a venture capitalist, and the venture capitalist funded most of this research. So building on that, I spun out a company in December, and we're going to use, we also have patents for these are safe crosslinkers. They're the first ones. We have patents for them in the use in hair. They make hydrogels. They do a bunch of stuff. So what I'm trying to do, and we've got an investor, they've already invested, the company exists. We're going to try and use stuff like hair, which is ironic for me, Paul, to fund the ALS research. And so I'm trying to fund it myself. I can't find a disease. The NIH never funded a single study on cyclic thiosulfonates. They were always happy to give me money to do basic biochemistry of this protein, but as soon as we made drugs, they never touched it again. So it's very hard to get. That's why they call it the Valley of Death, Paul.
Well, hopefully you survive. I have a question from Stephanie.
None of us survive, Paul. I study diseases. My probability of being alive is 94% right now. And in 25 years, it's about 30.
Well, thanks for that optimistic point of view, Jeff.
That's realistic.
Yeah. Stephanie has a question. It might have been covered. I missed the beginning. You said the drug is capable of crossing the BBB. Why do you have to administer intrathecally?
Yeah, so Stephanie, that's the right question. We don't. Our drug is orally bioavailable. What I was saying is that right now for SOD1 related ALS, there's only one treatment. It's called tofersen. It's an antisense oligonucleotide. They can only administer it twice a month because they got to inject it into the spinal cord. So they're doing intrathecal. We don't. And I really think where our compound ends up is as a co-therapy with tofersen, because in fact, the therapeutic index, the therapeutic window of tofersen is tiny. They are decreasing both. You have two copies of SOD1. It's dominantly inherited. ALS patients have a good copy and a bad copy. Tofersen decreases both SOD1. When you get to 37% of total SOD1, patients start to die. Our compound increases its activity. So we're going to eliminate, or not eliminate, but mitigate tofersen's biggest liability with something that's orally bioavailable and has better pharmacokinetics. I hope.
Great. Thanks, Jeff. Wealth of information in that statement. Thank you.
Yeah. Yeah.
Any other questions or comments? If not, I'd like to thank Jeff again for taking us on this tour of drug discovery program. It's interesting stuff. We look forward to seeing this actually have an impact someday.
Well, I want to thank everybody who came. I respect so many of the people on this list. It really means a lot.
Great. Thanks, Jeff. And just a reminder, we have a seminar next month. Philip Cowlick from Thiel about proteoform truncation and the the third annual International Top-Down Proteomics Symposium in Hanover in August is now open for registration. You can go to tdp2025.org and have a look there and get yourself signed up. Thanks again, everyone. Thank you, Jeff, and I hope you all have a nice day. Bye-bye.